ADCs have three basic parts: the antibody part, the cytotoxic chemo, and there's the linker that connects the two. First, the antibody part binds to the target on the surface of the cell. Antibodies can be designed to bind to proteins with a very high level of specificity, and that's what gives it the targeted portion. Then the whole thing gets taken up by the cell and broken down, which releases the chemotherapy part. Some sources will call this the payload or the warhead. That's the part that's attached to the heat-seeking part. That's what causes the cell death.
You're listening to the ONS podcast, where ONS Voices Talk Cancer, a resource from the Oncology Nursing Society. Through conversations with subject matter experts, we examine the important issues in oncology nursing, from new treatments to patient-centered research to advancements in clinical practice. Join us as we hear from nurses in all facets of oncology care, from bench to bedside and everywhere in between. Hello and welcome to the ONS podcast. I'm your host, Jaime Weimer, Manager of Oncology Nursing Practice at ONS. And today we're talking with Kenneth Tham, Clinical Pharmacist in General Oncology at the UW-Medicine in the Fred Hutchinson Cancer Center in Seattle, Washington, about antibody drug conjugates. This episode is part of a series about anti-cancer drug classes, which we'll include a link to in the episode notes. This episode provides free NCPD contact hours. To receive credit, please review the episode notes for any guest disclosures before listening. Following the episode, complete the evaluation we've linked in the episode notes. Welcome and thanks so much for joining us today, Kenneth.
Thanks for having me on.
So to start things off today, could you give us just a brief overview on the history of antibody drug conjugates in the field of oncology?
Absolutely. I'm really excited to be here talking about the antibody drug conjugates, or ADCs. To me, they have this really cool mechanism that brings together a lot of interesting developments in cancer treatment. And the idea of targeted chemotherapy treatment has been around almost longer than the use of chemotherapy itself. Paul Ehrlich, who won the Nobel Prize in Medicine in 1908, first came up with the concept of what he called a magic bullet, which in his mind was a treatment that could specifically target pathogenic cells while leaving normal cells untouched. Now, at the time, he was using this to refer to microbes in the context of infectious diseases. But it also laid the groundwork for the idea of a targeted, specific chemotherapy. Chemotherapy, by the way, was also a word coined by Ehrlich to describe the use of chemicals to treat disease, and then was later applied to cancer treatment. And cytotoxic chemotherapy was first used in the 1940s, and it was, and essentially still is, a cellular poison that affects all the cells throughout the body, not just cancer cells. That's why it's associated with so many side effects and why it's so important to get the dose just right. But because it was so toxic, the thought was, what if there's a way to direct the chemotherapy specifically to the cancer cells and leave the normal, healthy cells undamaged? Now, in the early days of chemotherapy, there really wasn't a good way to have this sort of specificity with most drugs before the advent of antibodies. Antibodies are these really huge, really complicated proteins. And so they have a level of specificity that small molecules just can't match because they can bind to their targets at so many different places. The first FDA-approved ADC was gemtuzumab ozogamicin, which was approved in 2000. This was an anti-CD33 antibody, which was gemtuzumab, that was attached to a cellular poison called calicheamycin. And in the last 25 years, there have been more than a dozen ADCs that have been FDA approved for various types of cancer, with many more still on the way.
Thank you so much for that brief overview, but really it does put it all into really fantastic perspective of how it's almost the accumulation of all the things we've tried and learned before, all the best parts of those things packaged together into one single treatment that can do all those things you've just said, really target, really be effective, but also spare the non-cancerous cells and the healthy cells in your body from suffering the consequences of those chemotherapies. So just really interesting to hear you describe it that way. So thank you for starting us off with that. So let's talk a little bit about the mechanism of action that ADCs are associated with. How do they work?
It kind of is a culmination of a lot of developments in cancer treatment. I think the mechanism of ADC is the really cool part, and what makes them so interesting and really exciting as they come through development. And sometimes why you'll hear them implied as quote unquote targeted chemotherapy. And so the ADCs have three basic parts. There is the antibody part, there's the cytotoxic chemo part, and there's the linker that connects the two. So first, the antibody part binds to the target on the surface of the cell. And as we mentioned, antibodies can be designed to bind to proteins with a very high level of specificity. And that's what gives it the targeted portion. Then the whole thing gets taken up by the cell and broken down, which releases the chemotherapy part. Some sources will call this the payload or the warhead. That's the part that's attached to essentially the heat-seeking part. And that's what causes the cell death. So as their name implies, ADCs have properties of both the antibody portion and the cytotoxic chemo portion. So besides just the heat-seeking targeted part of the complex, the antibody does more than just that. It doesn't just direct the payload to the cell. It can also initiate cell death, as most antibodies might. So any naked antibodies or antibodies that don't have that cytotoxic component, for example, through antibody-dependent cell toxicity. And so the mechanism of the action of the chemo itself really depends on what agent or what warhead is attached. Generally, they have some kind of cytotoxic mechanism related to many of the chemotherapies that we use in practice without attachment to the antibody. Some of them can be microtubule inhibitors, just like vinca alkaloids like vincristine. Some of them can be topoisomerase 1 inhibitors like irinotecan. Some can be alkylating agents and cause DNA breaks. So again, looking back at the arsenal that we have of cytotoxic chemo, these can all be incorporated into the ADCs.
Part of our conversation today will be encouraging and reminding nurses that when we're thinking of these drugs and we're providing them to our patients, it's not enough just to know it's an ADC. You have to know, well, what's it made up of? So this drug class, I think, is somewhat unique compared to some of the other drug classes we've covered in this series, in that the class itself, it's defined by sort of the structure of how these drugs are sort of created and linked together. But since those individual components, as you just mentioned, can be very different, after that similarity of the linking, we can have very different conversations depending on what specific drug, what antibody, what chemotherapy agent are we talking about. And so I think that will be the interesting part of our conversation today, is just keeping that top of mind. Now, you mentioned that we are referring to these somewhat as targeted drugs. And so we know that many targeted drugs do have a biomarker testing component to these. Do antibody drug conjugates require any sort of biomarker testing that nurses should anticipate looking for those results before somebody might be prescribed or become eligible to receive an ADC?
Intuitively, it makes sense that if you have a heat-seeking component, a targetable antibody, you want to make sure that you have the target available. And so for many of these agents, you might expect biomarker testing to confirm the presence of the target that the antibody needs to bind to. And this is true for many of these agents, not all of them, but a lot of them. One good example of this is trastuzumab deruxtecan. The trastuzumab is the antibody component, which is specific for HER2. You'll generally see this agent in HER2-positive cancers like breast cancer, gastric cancer, or lung cancer. These are just some of the cancers that can express HER2 on those cancer cells. In fact, trastuzumab deruxtecan was actually recently approved for any solid tumor with HER2 overexpression, regardless of the anatomic location of the cancer, and something that's called tumor-agnostic treatment. So you have the diagnosis of cancer, you identify the presence of HER2 overexpression on that cancer, and then you send in this antibody drug conjugate that seeks out HER2 and drops off the deruxtecan component, the cytotoxic component. This is also a really interesting case too, because trastuzumab deruxtecan was also recently approved in what are called HER2-low breast cancers. So these are breast cancers that have some expression of the HER2 target, but usually not to the same level that you would consider to be positive. And this is thought to be due to what's called the bystander effect. So the antibody drug portion guides the drug to the HER2 positive cells, but when the cytotoxic portion is released, it kills not only that cell, but nearby tumor cells that may not necessarily be HER2 positive. But again, in these cases, we are looking for the HER2 biomarker to send in the HER2-directed antibody. But not all ADCs require biomarker testing. So one good example of this is tisotumab vedotin, which is an ADC used in cervical cancer. The tisotumab portion is specific for a target which is called tissue factor. And the studies that looked at tisotumab vedotin, while they looked at tissue factor expression in an exploratory analysis, that is after the patients received the drug, they didn't specifically test for tissue factor when they were enrolling patients in this trial. So this agent, it's approved for recurrent or metastatic cervical cancer without the need to test for tissue factor expression. It's possible that patients who have higher tissue factor expression are those who respond best to this drug, but I think there's still more research that's needed before coming to that conclusion. And since those studies didn't specifically look for patients who had that overexpression, it can be used in all patients regardless of that expression. I also want to talk about another interesting case where the biomarker is being tested, but the biomarker isn't the target that you're looking for. And one good case of this is a newer agent that was approved that's called datopotamab deruxtecan. The datopotamab portion is specific to a target called TROP2, which is expressed on the surface of many epithelial cancers. This agent was first approved in hormone receptor positive, HER2 negative breast cancer, and then also recently received accelerated approval in patients with non-small cell lung cancer with an EGFR mutation, since those populations were the ones that responded best to the drug, datopotamab deruxtecan. So that's a little bit interesting. The antibody looks for a target, TROP2, but in both of these cases, in the breast cancer and the non-small cell lung cancer, you're testing for expression of different mutations or lack thereof. You're not looking for expression of TROP2. There's probably more research that needs to be done about the relationship between TROP2 expression and the presence or absence of these other biomarkers. But until we know more, we're actually testing for biomarkers that aren't the target of the ADC.
That really is fascinating. And as I'm listening, to me, it just reinforces the importance of knowing what drug you're giving, what are the requirements, what does the package insert say, but also it just reinforces that the main point when we're trying to test for and find biomarkers is to ultimately ensure we're giving the right drug to the right patient. And we're giving a drug to a patient that will be effective or potentially not especially harmful if we're looking at sort of pharmacogenomic biomarkers. But ultimately, and I think that's the part we have to make sure we translate to patients, is that we don't want to give you just any drug because it might be effective for somebody. We want it to be effective for you. Those are some of the conversations that I know we as nurses have. You know, a patient will come in and say, well, I saw this commercial for this drug and it sounds great. Can I have it? And sometimes the answer is no, because it's for a very specific indication or a very specific biomarker. And so that is important education for us to share with all of our patients. But as you've just described for some of these ADCs, it can be very complex on what we're looking for and why. And so thank you for stepping through some of those very unique cases in terms of what biomarkers we might see for certain ADCs that we are giving. So are there any primary like subclasses or subcategories of ADCs, or are they all very unique that we don't necessarily have any different buckets that we can kind of put them in?
It's kind of hard to put them into buckets, but I think one way that I like to categorize them is in terms of what's the target and what's the payload. So if you look at the name of these ADCs, there's always two parts. As you mentioned before, it's not just enough to think of its payload attached to an antibody, because those also have specific properties too. And so the two parts of the name, one part always ends in mab, which is the antibody, and the second part always refers to the cytotoxic payload. And sometimes if you can look at the mabs and the payloads, those will have similarities between them. So for example, there are two agents. One is trastuzumab deruxtecan, which I just talked about, and another one is trastuzumab emtansine. And these have the same antibody component, which is trastuzumab, but the anti-HER2 antibody. So we can expect them to have many of the same properties of anti-HER2 agents. So things like cardiotoxicity are associated with both of those, which require things like echocardiogram monitoring. Similarly, there's trastuzumab deruxtecan and datopotamab deruxtecan. And they both end in deruxtecan, which is the payload component. It refers to a compound known as DXD, which is a topoisomerase 1 inhibitor, just like irinotecan. I mean, they even sound similar, ending in tecan. So we know that the properties of the payloads will be similar, even though the targets are different. In this case, trastuzumab targeting HER2 and datopotamab targeting TROP2. There's a couple of newer agents that I wanted to talk about from the last year, and that's datopotamab deruxtecan and telisotuzumab vedotin. Datopotamab deruxtecan, as we mentioned before, is a TROP2-directed antibody attached to a topoisomerase 1 inhibitor that was approved in hormone receptor positive, HER2 negative metastatic breast cancer and EGFR mutant metastatic non-small cell lung cancer. The other agent is telisotuzumab vedotin, Telisotuzumab is a c-Met-directed antibody, and vedotin refers to a microtubule inhibitor called monomethyl auristatin E, or MMAE. And this agent was approved in metastatic non-squamous non-small cell lung cancer with c-Met overexpression.
Now, for the same reasons we just discussed, there may not be common toxicities across all of these agents, given that they can be very unique when compared side by side. But are there any common toxicities or adverse events that are notable? Or are there any individual agents that you would want to call special attention to?
I think there are common adverse events to antibodies in general and chemo in general. Stepping back a little bit, because we have both of these components, we want to watch out for the adverse effects of both of them. And so antibodies, as with most proteins, can trigger an immune response or an infusion reaction. And so many of the ADCs can also cause hypersensitivity or infusion reactions, although the rates of that are really variable and depend on the actual antibodies themselves. And then you have the cytotoxic component, the chemotherapy component, which has its own characteristic side effects. So if we think of really general chemo side effects, fatigue, nausea, bone marrow suppression, alopecia, these can be true of a lot of these ADCs as well. And then you'll get into some of the more specific class effects. So as you mentioned, Jamie, a lot of this depends on the specific type of antibody and the specific payload. So coming back to trastuzumab and emtansine, which again has trastuzumab, the HER2 antibody, and emtansine, which is a microtubule inhibitor. From the anti-HER2 component, you can expect cardiotoxicity, which is usually reversible reductions in ejection fraction, just like trastuzumab alone. And so these patients will often be on some kind of monitoring plan that involves regular echocardiograms. And then from the emtansine component, because it's a microtubule inhibitor, you'll see peripheral neuropathy. So you can often figure out properties of the whole drug by breaking it down into its components. And then you'll also see some adverse effects that arise from the combination. And this is where it gets a little bit more complicated. For me, at least, it's a lot trickier than breaking it down into its components. So for example, for trastuzumab and emtansine, there's a rare possibility of interstitial lung disease and pneumonitis. And this really isn't that common with trastuzumab or with microtubules inhibitors alone. And then if we look at trastuzumab deruxtecan, again, a HER2 antibody with a topoisomerase 1 inhibitor, the rates of ILD and pneumonitis can be really quite high as 10% even though trastuzumab and topoisomerase 1 inhibitors don't necessarily cause this as frequently individually.
Similar question, kind of similar expected response. Are there any unique pharmacology related strategies that we would use to manage or prevent the toxicities? Or is it kind of a similar answer where, depending on what each unique agent is, we would use those same strategies to help manage those toxicities if they were being given as single agent or as part of a different regimen? Or are there any truly unique strategies that we might use to help with the toxicity specific to an ADC?
Yeah, I think definitely there are strategies that are common to both the antibodies and the chemo, and then also those combination. So for example, in certain agents that are likely to cause infusion reactions from the antibody component, these agents might have pre-medications before every dose, things like antipyretics, antihistamines, steroids, and they might have a slower rate of infusion for the earlier doses to assess for tolerability. And then on the flip side, for cytotoxic chemo, we'd also use the same management strategies that we would use in general for chemo. So things like antiemetic premeds for prevention and treatment of chemotherapy-induced nausea and vomiting. Or if an agent were known to cause peripheral neuropathy, we might use the same strategies we might use in other chemo agents. So things like gabapentin or duloxetine, for example. But in addition to these strategies, there are other strategies for the more unique toxicities, especially ones that really have really high rates of toxicity that we wanted to have preventative management. So there's one example that I wanted to talk about, which was mirvetuximab soravtansine. This is used in ovarian, fallopian tube, and peritoneal cancers. And the mirvetuximab component is directed against folate receptor alpha, which actually has quite high rates of hypersensitivity reactions. And then soravtansine is another microtubule inhibitor, which can also cause peripheral neuropathy. But this particular combination actually has really high rates of ocular toxicity. And it's not really clear where this comes from because folate receptor alpha isn't typically expressed in the eye. So it's thought to be more due to the microtubule inhibitor, but again, not something that we typically see with that class of agents. So it could be the result of some property of the combination. The rates of ocular toxicity is quite high. The manufacturer reports that this can occur in up to 60% of patients even. So with rates so high, the manufacturer recommends a preventative strategy. So for this particular agent, for example, we'd recommend patients have required eye care. So ophthalmic steroids 6 times daily from the day before infusion to 4 days after, and then 4 times a day from days 5 through 8. And they also recommend artificial tears at least 4 times daily and as needed. And this ocular toxicity is something we do see in other ADCs that don't have the same target and don't necessarily have the same payload component. So for example, tisotumab vedotin and again, datopotamab deruxtecan can both cause ocular toxicities and both would have required ocular supportive care.
I'm so glad you brought up those examples because I remember hearing about the ocular toxicities for the first time and I thought, well, that is very bizarre. And again, as you said, not something that we really normally see with whether it's other antibodies or other chemotherapies. It's very unique. So interesting that it comes from sort of these combinations, but it's certainly notable and worth nurses understanding and exploring because it is something that we're not accustomed to seeing. And as you mentioned, some of those preventative or even management strategies can be pretty rigorous in terms of all the eye drops and things that we're just not as familiar working with our patients with. So thank you for bringing that one up. When it comes to the actual administration of these ADCs, are there any specific considerations, safe handling concerns that nurses should keep in mind as they're doing this administration?
The administration considerations are very similar to any kind of antibody or cytotoxic chemo component. From the antibody component, these agents will often have a short stability, so they often need to be made right before the patient is getting treatment. And again, since many antibodies generate an immune response, many of these agents may have slower rates for early infusions and then faster rates for subsequent infusions as tolerated. As we talked a little bit about before, many of these can have required pre-medications to prevent side effects such as infusion reactions or nausea. In terms of safe handling considerations, because they contain cytotoxic chemotherapy, ADCs should follow the same hazardous precautions as any other type of chemo. And so that means using appropriate personal protective equipment. It can mean using closed system transfer devices, depending on the institution, and making sure that medications are disposed of in appropriate waste containers.
What are some of the primary patient education points that we, as both nurses and pharmacists, should be communicating to our patients and their caregivers about ADCs?
Most of the patient education points are, again, related to the combination of the antibody and the payload. I think this can be somewhat daunting for patients, especially because it often lends itself to a long list of potential and unpredictable side effects. I think reiterating those potential side effects and, again, the supportive care treatment is really important. As you mentioned, that eye care for the ocular toxicity can be really burdensome for patients, but explaining why that's important and what the risk is that we're trying to mitigate is really important upfront. It's a lot easier to prevent toxicities, if possible, than trying to play catch up if the patient already has developed those toxicities. And since there are so many potential side effects, it's really important to communicate how and why the supportive care is done, and also when to communicate the provider or team to let them know that these things are happening if they do happen.
I think you bring up a really important point of basically having 2 drugs worth of potential adverse events and toxicities to try to communicate to our patients and then try to avoid it sounding like one of those pharmaceutical commercials where they rattle off like every horrible side effect that you can imagine. But by the end, you're like, why would anyone ever take this? We have to balance that. We certainly want to inform patients of the most likely or certainly the most serious ones that we need to make them aware of and so that they can tell us if they're experiencing these things. But it can easily overwhelm them, as you said. And so I do think that is sort of the art of nursing and of being a pharmacist, a clinical pharmacist working with patients is finding that sort of sweet spot of tell you just as much as you need to know the really important parts, focusing in on those, but also whether it's in written information or education that we provide, giving them information about some of those other rare ones, but still have to be informed. So it's that balancing act of giving them all the right information that they need, but not so much that it just pushes them right over the edge. It makes it sound scary because these are things that we can help them manage. And so it's that balancing act of not scaring them, but giving them the information they need to make the right decisions.
Absolutely. That's the art and science of healthcare.
Yes, for sure. So, Kenneth, how do you see pharmacists and oncology nurses working together and collaborating to help manage the adverse events and toxicities, provide this patient education, and really just serve as a trusted source of information for our shared patients, especially when they're receiving antibody drug conjugates?
I think as cancer care becomes increasingly complex and the agents that we use become increasingly more complicated, I think nurses and pharmacists can play an even larger role at helping manage these toxicities. And as we discussed, many of these agents have these required care schedules to help prevent toxicity. So counseling them on these, providing them with calendars. Jamie, you mentioned having some kind of physical or visual way to follow along can go a really long way in helping break this down into something that's digestible for our patients. And of course, breaking down everything into patient-friendly language can go a really long way towards developing trust. The way that these drugs work can feel very complex for patients. They're complex for us as healthcare professionals. And so helping them understand how the drugs work, why they're being used, can really help give them a sense of power and agency during a really difficult time. We might not know everything, but if we can explain why we're doing some things, that can really help patients feel like they're being taken care of. And of course, there's a lot of collaboration that can occur between nurses and pharmacists too, to identify the side effects, describe what we're looking at, the properties of the toxicities that they're experiencing, and also what can be done for them, sharing that these side effects, they're not necessarily unusual, but most importantly, there are strategies that we can do to help them feel better. And I think that goes a really long way.
So, Kenneth, what do you think the future looks like for these agents and their continued role in the landscape of cancer care?
Overall, I feel like the future is incredibly bright for these agents. There's only been around a dozen FDA-approved therapies. Despite this idea, the first agent came out in 2000. So 25 years later, there's only around a dozen FDA-approved treatments. But there are so many more that are coming through the pipeline. And as we've been discovering more and more biomarkers and developing more and more specialized antibodies, it's kind of only natural that more antibody drug conjugates would follow. You have all this complexity between the specificity of the antibody, itself a giant complicated protein. You have the chemical properties of the linker. There are some linkers that are being designed to be cleavable under certain conditions, such as certain pH environments, when exposed to certain types of light, for example. And then the properties of the actual chemo payload. And then you have the interaction of all these different parts. And so any change in any one of these properties can have really big changes in the drug's clinical effect. And then even more than that, I kind of want to talk about something that's pervading a lot of society today, and that's artificial intelligence. And of course, this is a big question mark in the world of healthcare today, but it has a lot of promise in the world of drug design. When you have a lot of data, a lot of different variables, that's where AI can really come into play. And so I mentioned that these agents are so complex and so elaborate, it can be really difficult to test these combinations one at a time. And so AI models can be used to sift through huge amounts of data to rapidly accelerate drug development and drug design to make drugs that meet specific criteria. And hopefully that can help researchers focus on the most promising agents and help get them to patients even faster.
Well, definitely very exciting. And I agree with you. I think lots of just really revolutionary things are on the horizon with all of these new tools, the use of AI. It sounds like we should get used to having ADCs as part of our sort of arsenal to help provide treatment for cancer. And they might only get more complex, but we'll be ready for them with wonderful podcasts like these to help provide that education for our nurses. Kenneth, thank you so much for joining us today on the podcast. It's been just great to have this discussion with you. I think these agents are so interesting and new for our nurses that are listening. And so I think they will really find this discussion valuable. As we reach the end of our episode today, we like to always run through a series of some just quick summary questions that help us to cover some of the key points that we've talked about. And so to start that off, how do you think healthcare professionals should evaluate any of their hidden or their implicit bias that they might hold for antibody drug conjugates?
I think that when it comes to ADCs, there may be this kind of implicit bias that this is such a new class of agents. Their mechanism of action is so sophisticated compared to other types of treatments, and the drugs themselves are so expensive that they must necessarily be, quote unquote, better than existing treatments. So better than antibodies alone or better than chemo alone. But better is such a subjective term. Do they lead to better outcomes, better toxicity? I think we can't just paint this with a blanket brush. We have to go back to the data and see how they were studied, see what they were compared to, what line of treatment they were in, and what toxicities were seen. I definitely think this is a really exciting class of medications, but we still have to evaluate them critically, just like any other treatment option, any new thing on the horizon.
I really love that. I think that's so true. What are some common misconceptions we might have about antibody drug conjugates?
Similarly to what I said before, I think when we think of ADCs as being targeted chemo, we think of these agents as having minimal side effects, or at least less side effects than other cytotoxic agents. And this seems to make intuitive sense because the whole idea is that the cytotoxic portion is being directed towards the cancer. But again, that may not necessarily be true. We have to see whether it's borne out in the data. Especially for many of the newer agents, these were studied in single-arm studies that weren't compared against cytotoxic chemo, or maybe weren't compared in the same line of treatment. And some of the older agents that have been around longer, even if they were studied against cytotoxic chemo, they haven't necessarily been shown to have fewer side effects. So yes, they may have different side effect profiles and different efficacy in different line of treatment. But again, it wouldn't necessarily be accurate to have a blanket statement and say that they're overall better tolerated compared to cytotoxic chemo.
What's something about this topic that's not often discussed, but you wish people knew more about?
One super exciting thing that I touched on earlier is how these agents represent a new frontier in precision medicine. So we talked earlier about biomarker testing, but many of these agents are being studied in tumor agnostic trials, or what are called basket trials. And so in these trials, regardless of whether patients have breast cancer, lung cancer, colon cancer, what have you, if that cancer expresses the relevant biomarker, they go into this basket, this big basket for study with these agents. And so there aren't too many tumor agnostic treatments that are approved by the FDA, but one of them is an ADC. We talked about this earlier, trastuzumab deruxtecan, which is approved for any solid tumor with HER2 overexpression without any good alternatives. So as our understanding of precision medicine, biomarkers, and these drugs becomes more refined, we may start to see them being used across a wide variety of tumor types.
What additional training or education do oncology nurses need to stay current on this topic?
Yeah, it can be really daunting to keep up with this as a whole class. We talked so much about how every agent has kind of its own properties and its own differences. Structurally, mechanistically, there's similar at a very high level, but when you really drill down to it, they can be quite different. And they're used in different disease states and can have very different toxicities. And there are so many more agents coming down the pipeline. So I think nurses can continue to keep up with new ADCs as they're approved, recognizing that they have their own unique characteristics, but using the sort of framework to think on how to think about the ADCs. I also think that keeping up with some of the newer antibodies can be really helpful because many of the ADCs are based off of existing antibodies. So if you already know the properties of an individual antibody, then if an ADC comes to market, many, many years later, it'll help be a shortcut to understanding that ADC. You'll already know some of the properties of that agent.
And finally, are there any additional resources that you would recommend for patients or even providers who want to learn more?
Yeah, I think because these drugs are so new, there's not a whole lot of data out there. It can be really hard to have a lot of personal experience with these because they can be so new and some of them may not be used in very common disease states or common biomarkers. So for any particular agent, I always like referring to the package insert and also the primary literature that led to its approval. I think this is just good to have a baseline understanding for every new drug that comes out. For patients, I actually think NCI's website is a good, reliable source. It can be a little big and daunting and unwieldy, but it does a really good job of breaking these concepts down into patient-friendly language and even shows how to pronounce some of these more elaborate names. And then for providers, I think resources like this podcast for one, but also any other resources produced by national organizations like ONS, HOPA, and CODA. These are always a great resource. I love the patient education sheets that are put out by ONS, HOPA, and CODA, and ACCC. I think they're really comprehensive and can really standardize the language that's used around a lot of these side effects.
Absolutely. I couldn't agree more. I'm so glad you mentioned those. Those are a wonderful resource that our organizations collaborate together incorporating what we've talked about today, that shared perspective of both nurses and pharmacists to help build an education document that can serve the patient best. So a great resource, both for ADCs and many other drugs and drug regimens that we provide here in oncology. Well, Kenneth, thank you again so much for joining me today on the podcast. It's been a great discussion. I've learned a lot, and I know that our listeners will really enjoy what we've talked about today. Do you have any final comments that you'd like to share with our listeners?
Yeah, I just want to share that you could probably tell from the way I'm talking. I find this a really fascinating, diverse class of medications. It really builds off of what we know about antibodies and cytotoxic chemo individually. And I'm really excited to see this field continue to develop and what else might come of it. I'd like to thank you for your time, Jamie, and thank you to the listeners of this episode.
Wonderful. Well, thank you, Kenneth. Thank you for listening to the ONS podcast. Tell us about your favorite part in this episode by leaving a review wherever you download your podcast. For more resources and information about oncology nursing, visit us at ons.org and ons.org/voice. The ideas and opinions shared in this episode represent those of the guest and not necessarily ONS.